Hi, I am Krzysztof Mikołajczak from the Institute of Plant Genetics, Polish Academy of Sciences in Poznań. I've got opportunity to conduct my phenotyping experiment within fifth call of EPPN 2020 Transnational Access. The plant phenomics installation was provided by Alsia Metapontum Agrobios Research Center located in Basilicata in South Italy. Project entitled High Throughput Phenotyping of Barley Varieties Response to Drought for Understanding Crops Adaptation to Arid Climate was aimed to characterize the variation and dynamics of growth and development of a spring barley collection exposed to water scarcity. A secondary objective was to identify an association between the traits measured during the time course of experiment and post-harvest yield components. Plant material consisted of 120 spring barley accessions. Each genotype was represented by 4 replications. Altogether, 480 pods were used in the experiment. Soil moisture was controlled by target weight-based watering using automated system on the platform. Initially, plants were grown under well-watered conditions, above 80% of field capacity, until tillering stage. Next, the irrigation was stopped and water shortage was maintained for 3 weeks to reach and hold that 20% of FC, and then plants were rewatered. Pods were labeled by special barcode stickers detectable by Lemnotec sensors. Imaging started few days before drought application, and it was continued during stress and after rewatering. Each plant was imaged sequentially employing top and side view camera to capture images daily that covered the visible light spectrum, near infrared, and fluorocam was also used. This provided information about plant height and growth dynamics, shape of the plant, projected shoot area, shoot biomass, color distribution, and water use efficiency across barley collection. Lastly, plants were moved to non-automated greenhouse for cultivation until ripening. Thereafter, spikes were collected for post-harvest yield components estimation. EPPN 2020 allowed me to carry out the high-throughput imaging of barley plants, which is technologically impossible to implement in my country. Secondly, thanks to EPPN 2020, I met a great people, qualified researchers, and our bilateral cooperation is continued within a new project that has been currently started. Unfortunately, I was not able to visit the access provider and participate in the EPPN experiment directly due to the COVID crisis. So, the more, I would like to thank the whole team in Metaponto for their work and effort to conduct the experiment successfully. Many thanks, guys.